I don't think I can sell it anywhere.
Okay. All right. I'd like to introduce Ken Burns and John and Mike.
Okay. Well, welcome again, Bert. We're delighted to have Dr. Flanegan here. He is the longtime, I say that advisedly, chair of the Department of Biochemistry and Molecular Biology in the UF College of Medicine. He is a senior virologist working on the coronaviruses, which includes polio and hepatitis C and all this stuff, more or less. And so Bert is going to talk to us about hepatitis C. Bert received his master's degree from FSU, and that was good enough to get him a commission in the Navy during Vietnam. And it was good enough actually to get him posted to Annapolis as a professor teaching chemistry to the midshipmen. So he spent his Vietnam War being saluted every day by the midshipmen. So that was kind of an interesting experience. Anyway, after they let him out finally, he went to the University of Michigan, where he received his PhD. And from there, he moved to the lab of David Baltimore at MIT to do a postdoctoral fellowship. I'll say that Bert has had good taste in some of his associations. He's worked with two Nobel laureates, David Baltimore, who discovered reverse transcriptase, and he did a sabbatical with Tom Check at the University of Colorado, working on actually enzymes that contain RNA. So Check was experience on her. We weren't sure Bert was going to come back from Colorado, but he did eventually show up. And so, yeah, but subsequently, he's also done a lot of collaborative work with a man named Charles Rice, who's a professor at the Rockefeller University and one of the recipients of the 2020 Nobel Prize in Medicine and Physiology. I think Harvey Alter was the second person. I can't quite remember the third person was Bert, but I'm sure you'll tell us. So without further ado, I'll let Bert speak for himself. Welcome again.
Okay, thanks, Ken. I'm happy to have the chance to be able to do this. I haven't really given a talk like this particular talk, and so I put something together, and I hope that I can make it clear. And really, I'll go ahead and try to share my screen. Let's see if I can get the right one. There we go. Okay.
Mike.
To me, is that okay? Can everybody see that?
Yes, it looks good.
Okay. The talk is in my field, although I have worked on hepatitis C in my lab I did for a period of time, really right after some of the really important work was done by Charlie Rice in terms of finding an infectious clone for the virus, which really opened up the whole field in terms of doing genetics and biochemistry with the virus. So I've known Charlie since those days, and this is, basically a virus that we have really good drugs for now, but it's still a big health problem. So the other three winners for the prize, the first Harvey Alter is shown here. And he really made the initial discovery regarding what now is known as hepatitis C virus, but basically really showed that there was something in human sera that was not positive for either hepatitis A or hepatitis B, and that it was transmissible. And so he basically, they didn't have a real good name for it because they couldn't really identify exactly what it was, but they called it non-A, non-B hepatitis. And so we had, and that was a huge problem because we had developed good screening for hepatitis A and hepatitis B in the blood supply, but we really didn't have a test that was available to test for non-A, non-B hepatitis. So the second person to win is Michael Houghton, and Michael and his team basically came up with a first Cdna clone of the infectious agent, and that was although it was not an infectious clone, that was left for Charlie to do, but it was a small piece of the RNA which they used as a starting point, and then basically walked and really could then get more of the sequence from that starting point in the RNA genome. But this was a big deal because it eventually led to testing and assays that we could detect hepatitis C. So it was, identified as a virus, the infectious agent. It was renamed to be hepatitis C, and it was found to be in what's known as the flavivirus family. And so that was a major discovery. And after that, the goal was to try to get an infectious cDNA clone, which will allow you to do a lot of additional important experiments in terms of learning more about how the virus replicates and developing antiviral drugs, which eventually happened. And a lot of that was based on Charlie Rice's work. Okay, so that sort of sets the starting point. And so I've kind of gone through this up here in terms of the three major contributions in this, and really the first cDNA clone resulted, as I was saying, in sensitive tests, because we then could make antigens or viral proteins, which could then we could develop immunological based tests for looking for the virus. And then eventually what Charlie Rice did in terms of having an infectious clone led to the development of effective antiviral drugs. In fact, these are really, really good, and they actually clear the virus. So it's one of the best stories we have in terms of antiviral drugs for viruses. Okay, and so just a little bit more about hepatitis C in terms of the real basics. So we have hepatitis A, B, and C. There are also a few others, but these are the big players that we're involved with. So hepatitis A is actually picornavirus, the type of viruses that I've spent most of my career working on. So that picornaviruses includes polio and Coxsackie virus and some others, or many others actually, and then the human rhinoviruses. So it's hepatitis A is in that basic family of viruses, but it causes acute infection. So you can get infected and have hepatitis, but then you get over it and the virus is gone. Sometimes, especially in older individuals, it can be quite serious in terms of the infection. Hepatitis B and hepatitis C is transmitted through the blood, and both of these viruses end up causing chronic infections, which can lead to additional, liver damage and cancer. So they're more of a problem. And then in addition to the chronic in addition to the acute infection, the hepatitis B and hepatitis C actually can cause chronic infections where the virus is still there, is still replicating, so the person is still infectious, can transmit the virus to others, and eventually that can lead to hepatocellular carcinoma. The thing about hepatitis B is we do have a good vaccine for it, and so that prevents, is very effective at preventing transmission of hepatitis B. Hepatitis C, we have good drugs now, but we do not have a vaccine, and the drugs are pretty expensive, and so the drugs really work well, but in terms of really getting rid of a virus, in terms of a major disease, the vaccine is always the best way to go. So that gives you a little background on in hepatitis, and a little bit more in the area of hepatitis C. It's in what's known as the flavivirus family, and in a subgroup, hepatitis C is hepatocity virus family, and it causes hepatitis. Transmitted by contaminated blood and sexual transmission as well. It's all over, distributed around the world. There's no seasonal incidents. The biggest risk factor are people who use IV drugs illegally and reuse needles, and healthcare workers who might get a needle stick. We do not have a vaccine, but there are multiple FDA-approved antiviral drugs that actually work well. So hepatitis C, normally the acute infection is pretty mild, but almost 80% of people 70 to 80% of people who get infected in the first place develop a chronic infection. And a lot of times you don't even know you're infected. The symptoms are so mild for a long time. And then gradually the inflammation in the liver gets worse, and you start to have symptoms consistent with hepatitis in terms of inflammation in the liver. This particular slide notes that we really, prior to the drugs, there were 130 to 150 million people infected worldwide, so it's a huge problem. The new drugs have worked really pretty well, and so the current numbers are about half that, or estimated to be about half that. So that's clearly made a difference in terms of reducing the number of infections, which also reduces the transmission of the virus. So in particular, pregnant women, if they have a hepatitis C chronic infection, they could pass it on to their kids, at birth, and so that gets into a very difficult cycle of transmission of the virus. So I know you're not here to be in my virology class, but I do want to, or maybe you do, but I do have to give you a little background to kind of set this up in terms of talking about what Charlie Rice did. So this diagram is a simple diagram of the replication strategy for different RNA viruses. And we're not going to go through all of this. I'm going to focus on the plus strand RNA viruses, and really on this one particular diagram that I'm pointing to. And in this, this is basically the strategy for replication of the flaviviruses and the picornaviruses. So these viruses, they have a one piece of RNA. It's single-stranded, and it's one strand of RNA is packaged in each virus particle. And when we say it's plus-stranded, that means that it has the polarity, if you will, to be translated into proteins. So it's basically all you need to start the infection is really just the RNA. The protein coat and the membrane coat on these viruses is really there just to help protect the RNA and to help the RNA get into the cell. So this positive strand RNA is used to make the viral proteins, and it's also goes through a replication cycle. In terms of the overall strategy, it's pretty simple. You have this positive polarity RNA, it's like a messenger RNA. There's in the replication cycle, the virus encodes an RNA-dependent RNA polymerase that copies the RNA from one end to the other. So it makes a complete minus strand copy of the genomic RNA, and then that minus strand is copied back into plus strand RNA. And so that's where the action is at in terms of the virus making new copies of itself. It has to make a lot more. In other words, this product is basically identical to what comes out of the virus in the first place. So you get a lot of new plus strand RNA, which is then packaged into virus particles. And this process is really quite efficient in infected cells with both the coronaviruses, for example, poliovirus, Coxsackie virus. And actually, I didn't bring it up, but actually our friend, the COVID-19 virus, SARS-CoV-2, is in this group as well. It's a single-stranded plus strand RNA virus. And so it's in this particular category. Anyway, these viruses replicate well. They make a lot of new plus strand RNA. They make a lot of viral protein. And overall, they make a lot of new virus particles. And then those new virus particles go to infect other cells, and then eventually, are transmitted to other people, other host animals, and cause other infections. So you have to kind of understand that to basically, at least at that level, to fully appreciate the work that Charlie Rice did in coming up with the infectious clone. So let's look at this type of virus in general, and not to go into too many details, but there are some other virus members here that you'll recognize. Yellow fever virus. So I think probably most people in the audience recognize that as being a virus that's been around a long time, caused a lot of trouble. Fortunately, we have a vaccine for that. Dengue virus is all over South America, Central America, and inching closer and closer, and being brought into the U.S. West Nile virus is another member in this group, and as we all know, is already in the U.S. And causing some problems in the U.S. It didn't used to be here, but it's here now. And then we have hepatitis C virus. So these are all viruses with a similar genomic arrangement and structure. So if you can see them in the electron microscope, their picture, these round circular particles or virus, the flavivirus particles. And this is a cartoon over here showing that the RNA, the single piece of RNA, is packaged inside of the virus particle, and then you have the viral proteins, different ones that form the capsid, along with a membrane envelope that surrounds the outer part of the virus. So the envelope is just, in terms of general, I never can pass up this opportunity in talking about virology, because this is, coronavirus, which we're all worrying about right now, also has a membrane envelope on its outer surface. And this is the same for hepatitis C. It's not true, though, for polio and hepatitis A. that membrane makes the virus sensitive. Once it dries out, that membrane tends to shrink and change its shape. And it's very sensitive to anything that will affect membrane structure. So anything, an organic solvent, any kind of alcohol, detergent, for example, really disrupts that membrane. And that's why just washing, your hands is so effective in terms of really basically inactivating these type of viruses. So it's really good relative to inactivating hepatitis C. So it's not so good. It's not that stable in the environment. And certainly just alcohol and, hands and washing your hands basically would inactivate most of that virus. Okay, so now how does it work? How does it do its thing in terms of replicating, making its proteins? So this is obviously looks kind of complicated, but I just have a couple of take-home messages here. And so basically the single RNA, which is inside of the virus, it basically functions like a messenger RNA. And a messenger RNA is the type of nucleic acid that we make in all of our cells. It's copied off of DNA, our own DNA in the cell, and then it goes into the cytoplasm of the cell and it gets translated to make the proteins that we have in our cells. And so it's meant to be, that's one of its primary missions. There's a lot of other functions for RNA as well, but encoding the viral, the cellular proteins is a primary function. The RNA is, compared to DNA, is not very stable. So RNA turns over pretty fast. And so it makes it, susceptible to degradation. One of the reasons the RNA viruses compared to DNA viruses, that's one of their, areas where they are more likely to get degraded. And that's why the outer capsid is so important in protecting the RNA. One way they get around that is they just replicate so well and so fast that they make a lot of new progeny virus. Anyway, this RNA, without going into the details, gets translated and then to make one long protein, which is shown here. And this protein makes enzymes that are called proteases, which are like protein scissors. They can cut this RNA, they can cut the protein into different sites. And so when that's all finished, there are basically a couple of proteases that the virus encodes, and there's another one from the cell, and that cuts all of the capsid proteins and what we call the non-structural proteins, or the replication proteins. These are the proteins that make the RNA replicate. And so we get a whole collection of these proteins inside of the cell. And so they basically replicate the RNA and end up making additional virus. So in order to get a clone, and we're going to keep coming back to that idea, a cDNA clone of the virus that really is functional and infectious, so we can do these kinds of studies. That is, we can take the RNA and put it into cells and make these proteins and then find out what they do in the replication cycle. It's important that this process will actually work in the infected cell or infected animal. And basically this is the heart of it right here in terms of that working. If anybody has any questions as we go along, I'm happy to, answer. So if you just give me a shout out or, let me know, I'll be happy to.
So
talk.
the little arrows without a curved line means that they use the enzymes from the cell to break that up. I see. Okay.
Yeah.
Good. And the other ones, they produce their own enzymes to break it up.
So what they can do is they can basically this was a major question at one time, and that is, how do these proteases that are built into a precursor protein actually cleave other proteins? So the initial, like this particular event with this NS2 protein can actually occur when the precursor protein folds. It basically folds in such a way that the cleavage site folds into the active site of the enzyme. And so that's an autocatalytic cleavage event. And so that cleavage will occur, just automatically when this protein is made.
That's amazing.
Likewise, this NS3 protein can get started the same way. And then after they're out, then they can carry out additional, cleavage events by working as an individual protein. So this protein, the NS3 protein on its own can then go, do these cleavage events in what you'd call a trans cleavage. Yeah, so they're basically, they've evolved that way, so they're ready to go.
I have a question related to the mutations. Do we see this genome getting mutated a lot, or is it not too many mutations? Do you see that?
So that's really a really important question, and I'm gonna get into that pretty heavy duty here because it's an important issue.
Okay, thank you.
Relative to this. And so basically the next slide. So basically the generation of RNA virus quasi-species. And the point here is, and I think most of us are hearing in the news every night about new variants for coronaviruses. And the SARS-CoV-2, there's a new variant, the UK variant and the Brazil variant. Well, these are just mutations that occurred naturally in coronavirus. And I told you earlier, coronavirus is the same type of virus, genetically. It's a positive strand RNA virus. And so these viruses, the polymerase has a high mutation rate compared to DNA polymerases, and this is actually very high mutation rate compared to DNA polymerases, which copy our DNA in our cells. But that's okay for the virus because they make a lot of copies of themselves. So when you consider a rate like this is 1 in 10,000 nucleotides copied is a mistake. And so in hepatitis C, the genome is about 10,000 nucleotides long. And so every time that virus replicates, you're getting at least one new mutation put into the genome. And so basically over time, even if you started with one molecule or one virus particle, you're going to end up with a lot of mutant genomes in the host. And so basically that's any virus in this group ends up generating a lot of mutants as it replicates. So it's it's not really a species. That's why they call it a quasi-species or a mutant swarm. So basically these viruses are complex distributions of mutants or mutant swarms or quasi-species. So these changes in the genome, they can either be totally negative, and be selected against, so that might be something that would make the virus no longer infectious. So you would select against that. Or it may be negative, have a deleterious effect, but may not kill the virus. So it would slow it down. And so that would be, not as favorable for the virus. It could be totally neutral and not have any effect one way or the other in terms of how the virus replicates. And so that would happen, but it wouldn't be selected for either way. So it would just, get diluted out with time. So neutral mutations don't really, they happen all the time, but they don't really have any impact because they're not being selected for or against. And then the ones that have a positive advantage or give the virus a positive advantage, they're selected for. And so the virus is replicated long enough where it's really has a pretty optimal genome as it is because, it's gone through a lot of selection and evolution over time, but there are changes that can occur when the environment changes to help the virus replicate better. So this concept of the quasi-species, it basically allows RNA viruses to rapidly adapt to new situations and are critical for survival of the RNA viruses. And this was one of the problems that was so hard to get a infectious clone for hepatitis C, because it basically is, existing as a quasi-species. So this is this slide. Actually, there's an important point here I want to really make. So this shows this development of the quasi-species. So one virus particle, infecting a person and generating a bunch of new progeny virus. Basically, you start with the sequence, that the virus had when it started, and then as it replicates, mutations occur at different parts of the genome. Some of these are neutral. A lot of them are neutral. Some of them are, could be lethal and actually totally kill the virus, and others could potentially, be an advantage to the virus and be selected for. So the ones that, that are basically advantageous are selected for, and they become a predominant part of the virus. And so, although we have all these mutations in the population, there is an overall sequence for the viral RNA. It's called the consensus sequence. And that is, if you look at each nucleotide position in the viral genome, it is the nucleotide at that position that is predominant. It's the predominant nucleotide at that position. So, there are 4 different nucleotides that make up the RNA, A and T and G and C. And so whatever works best there eventually gets selected for, and then that's going to be, the predominant sequence. So you can see what this sequence at the bottom here is called the consensus sequence. And you can just see, for example, if you look at this little square box, if you just kind of, eyeball this and look up, you can see most of the individual sequences, contain that particular mutation at that site. So that was a positive, had a positive effect, and it was fixed into the consensus sequence of the virus. So this is really sort of the heart of what Charlie Rice did and won the Nobel Prize for, because he recognized this issue and he built his clone so that it would have this consensus sequence. In other words, if you've got the best nucleotide at every position in the whole viral genome, you would think that would be the one that's going to replicate the best, or replicate at a level that you could actually measure it. And so that was his goal, is to get a consensus, to get a clone that had the consensus sequence in it. Okay, is everybody okay with that? Because that's a critical point in terms of, knowing what he was trying to do. And this is more, this is complicated, looking at all this and trying to figure out what's really going on. And it is complicated. Okay, so basically that, what I want to do now is really focus on this component here, the work that Charlie Rice did in terms of coming up with a DNA clone that's, basically a plasma DNA that you can carry in a bacteria, but contains the sequence for the virus. And so basically the viral RNA is shown here, and if it has the consensus sequence, it should be able to replicate in a host animal. And so that's what Charlie Rice did. And basically, you would have thought the work that was done by Michael Houghton, and that was done in 1989. And the work it took till 1997 before Charlie Rice had an infectious cDNA clone. So they were real close, but it took a long time to get there, and a lot of people were working hard trying to do that. So I'm going to tell you a little bit about that in terms of what his thinking was in terms of doing that. So here's a picture of Charlie, and here's a electron micrograph of hepatitis C, and it's a big, compared to the coronaviruses, it's a little bit bigger. It's got a bigger genome, and it's 50 nanometers in diameter. And so you can, easily see the virus looking in the electron microscope, and you can, measure the size accurately. And as pointed out here, there's still, even with good drugs available, we still have about 70 million people infected worldwide. And the problem is a lot of those folks don't even know that they are infected with the virus because it's a chronic infection, and they may not even have any symptoms yet. And so, that's a silent, stays in, this, early stage for quite a long time. It can be years. Some people have had the virus 10 years and they don't even, have any real symptoms yet. But eventually, once it gets started, then it really starts getting serious because of the cirrhosis of the liver, which then can lead to liver cancer. And it's one of the primary causes that, reasons that people have to have liver transplants if they have a chronic infection with Hep C that's not treated. Okay, so here's a diagram of what goes on in my lab all the time, and this is and so this is a diagram that was put together by Vince Raccanella at Columbia, and he basically was the person who created the first infectious cDNA clone for polio. And so, but the idea is basically the same for hepatitis C. So the idea is you want to take the RNA, the plus-strand RNA that comes out of the virus, and you want to take that and you want to copy it into DNA. And I won't go through the details of that, but it's a straightforward process now to copy RNA into DNA. And actually my old boss at MIT got the Nobel Prize for that in discovering reverse transcriptase in terms of the enzyme that copies RNA into DNA. And then putting it into a capturing, one molecule of that cDNA, which you then turn into double-stranded DNA in a plasmid that can go into a bacteria. And then you can carry it and amplify that DNA in the bacteria. So this is the bacterial plasmid containing the poliovirus sequence. You can set these up where you can basically transcribe and turn it back into RNA, so you can make in the test tube in the lab easily turn it transcribe and make more plus-strand RNA, and then you can take that RNA and put it onto cells if the virus, if the virus replicates well on cells, and you know what that cell culture system is, you can transfect the RNA directly into those cells, and the virus will start replicating in those cells. And then when it replicates in the cells, it actually will produce new virus. And so you'll make, millions, thousands, depending on how many cells you got, you make a lot of new virus in the cells that are growing, in culture that have been transfected with the transcript RNAs. If this is a true infectious copy of the virus, and then copy it and faithfully, so that you have an accurate copy of the RNA that will go into plasmid, and then you can make infectious RNA that you can put into the cells. Let me point out, let's go back just 1 second and think about this, especially, it looks easy, but remember the virus, the RNA that's being made in cells and getting packaged into virus particles, a lot of it has mutations in it. And so are you going to get one of these that are, have mutations that don't work, or get one that is a sequence that works well? So, there's a lot of copies of the RNA, and then when you clone, you get a subset of that. And so it's kind of random chance whether you're getting one that is just or not.
So when it's in the cDNA form, is it dangerous?
If it's in the double strand, that would be single-stranded, but I mean, usually that's just immediately turned into double-stranded RNA. So the double-stranded RNA, in principle, is dangerous and, in fact, is infectious. So in the case of the poliovirus clone that was done by Vince Raccanella, basically he put the viral sequence into a plasmid DNA, and he showed if he transfected that plasmid DNA directly in cells, it actually ended up producing new virus. So the RNA was transcribed off of that DNA and then was infectious. Even though it didn't have the right setup to do that, the cell figured out how to make that RNA off of that sequence in the cells. So yes, that's considered hazardous, and you got to treat it, like an infectious agent. I mean, normally it wouldn't be very infectious because it's a nucleic acid. It can't, get into cells all that well. But, I think now, if we think about just as an aside, basically with the Moderna and the Pfizer vaccines, we're taking a piece of messenger RNA that codes for only one viral protein, the spike protein on a coronavirus. And they wrap it up in a membrane envelope, and then they throw it on. I mean, that's what they're putting in your arm, a piece of RNA wrapped in a membrane envelope. And that goes and gets into cells. And it starts to translate and make those proteins. And of course, it only has the coding capacity for one protein, so it makes the spike protein. But then that spike protein, then the immune system sees it, and that's why you get an immune response to it. And it's working amazingly well. And the RNA in that vaccine is gone in 24 hours. RNA turns over fast, so it has to get in fast and get going in terms of what it's supposed to do. And so this, is pretty efficient, but it's not nearly as efficient as the virus is. The virus has evolved this fancy delivery mechanism, if you will, to bind to the surface of the cell and help the RNA get in the cell. So it can get in, it's just not quite as efficient as the virus itself.
Thanks.
All right, so basically what Charlie wanted to do was what we've been talking about, make a infectious clone where he could transcribe the RNA, the viral RNA off the clone, and then put it into a host animal and show that it was infectious. Now, you want, and what they really wanted was a cell culture system where they could put the RNA and look, follow its replication, like I was demonstrating in the previous slide. But at that time, there was no cell culture system for hepatitis C. So they had not only a virus that rapidly generates a lot of mutants, and it was a bigger virus than polio. Polio is like 7,500 nucleotides, and hepatitis C is almost 10,000, so it's a bigger genome, and then no cell culture system. So basically, they were kind of handicapped right from the beginning, but took this project on anyway. So the first point here, I mentioned before, the Cdna clone of HCV was first isolated by Michael Houghton and his collaborators in '89. This was not a full-length copy, but it was. They ended up making, a clone that contained most of the viral genome, but it wasn't infectious. But it was really valuable, and that's why he got the prize. It helped him, identification of what the infectious agent was. It was hepatitis C. It was a member of the flavivirus family, and it allowed them to develop new assays for testing for the presence. And so that was a major advance. And plus, you could express some of the viral enzymes, the proteases, the polymerase, and so forth, that they had isolated. So that allowed a lot of research to move forward. And then Charlie and others took on this, challenge and wanted to, thought they could move this forward pretty fast to get an infectious clone. It had already been done. He had actually done it for yellow fever already in his lab. And then there was already infectious clones for polio. So, it seemed like, okay, this is pretty straightforward, should be able to do this. And so basically he started out by isolating the virion RNA from the H77 strain of hepatitis C virus. This was isolated from human sera. And this was used to prepare. So basically what they did is they made what's called the Cdna clone that came up when we were talking before. You take reverse transcriptase, you copy the RNA into DNA, and then it copies the DNA, single-stranded DNA gets copied into double-stranded DNA, and then you can clone that. Cdna clones. So very similar to what was done with polio, but this is a much bigger genome. So they had a quality control process in terms of using restriction enzyme digestion and looking for certain proteins to be made. And from the 200 clones they had, they selected 34 clones as good candidates to test for infectivity. They thought, okay, well, if we're lucky, this is going to work right off the bat. And, these look like they should be full length. But the problem was, again, they didn't have a good way to test it. So the only way to do it was to directly inject the RNA into the liver of chimpanzees. That was the only animal model there was for following hepatitis C virus replication. And so, but it turned out that none of those clones were infectious. So it was clear that this was going to be a much harder job than originally thought. And so they had to step back and start thinking about this. So what they did was and sequencing wasn't as fast and as easy as it is now. If you have a sample something you want sequenced, you send it off to a company and you send your sample in, and a few days back they'll send you an email that has your sequence in it. But in those days, most people were doing it themselves, and it took quite a while. And so those were the old days where we had these big, sequencing gels and apparatus that we did in our labs. Anyway, 6 of the above clones that were out of the 34 were picked for sequencing from end to end. And then they basically figured out from those sequences what the consensus sequence was. In other words, at every nucleotide position in those 6 clones, they figured out what was the predominant sequence at that point. So that told them what the consensus sequence was for the HCV genome. And then when they looked at the sequence in the individual clones, they realized there were significant variations from the consensus sequence itself. And so all of them had individual mutants that made them different than the consensus sequence different places across the genome. So this explained why none of the clones that they had tested before were infectious, because none of them had the consensus sequence. So that meant they were going to have to go back and figure out how to make a clone that had the complete consensus sequence. And so that's where they went next. So so based on that, the goal was to construct a full-length cDNA clone containing the HCV consensus sequence. So they had these 6 clones that they had the sequence of, and they, I guess, looked at that long and hard, and they figured out, I mean, it's DNA now, and so you can cut DNA at specific sites with enzymes that are called restriction enzymes. And one of Ken's good friends at Hopkins discovered that, and those types of enzymes. And so there's a whole bunch of them now that have been isolated, and you just buy them. And so they could cut the clones up into pieces, and they picked 28. It was about, I counted it fast myself looking at his paper. It was around 28 or so restriction fragments that they picked from the 6 sequence cDNA clones, and if they put that together like a puzzle, they could put it together in such a way that they would have the correct consensus sequence. You get 1 fragment from one clone, and then 2 more from the other one, and 5 from this one, and you put it all together and ligate it all back together. And so it was really a pretty, certainly considering the techniques available in those days, it was a major task, to undertake. And so they basically constructed, a clone with the consensus sequence. And then they also saw some variations in the sequence at the 3' and 5' ends. And so they made a few additional clones to see if that made any difference in terms of it being infectious. And then this was the final thing. They wanted to test it for infectivity. That's where all the money was at, and all the, possibility for the future in terms of using these. So this was the paper that they published, where in fact, well, we wouldn't be here, talking about this today, and Charlie wouldn't be a Nobel Prize winner if it hadn't worked. But he published a paper in Science in July 1997, showing that basically he had isolated the first infectious cDNA clone for hepatitis C, and that it would replicate in chimpanzees. So, and then we'll talk about a little bit of the information in that paper. Just got just not much time left, but basically he created this. This is a diagram showing the 5', 3' ends of the infectious clone. It turned out to be infectious, but this was the consensus clone. And then he made some variations of that by adding some different nucleotides at the 5' end, and changing some of the sequences at the 3' end. There's a poly UC track near the 3' end, and that varied some in the different clones. So he added those on, and then he tested all of these for infectivity. And I just wanted to add a personal note here, because a few days or just a few weeks before that paper was published, there was a meeting of the American Society for Virology in Montana. And basically I went to that meeting, and when Ken asked me to do this talk, I said, oh yeah, well, I heard Charlie give the talk at the ASV where he described his results, and we all knew that, well, we didn't know if he was going to win the Nobel Prize, but we knew he was going to get some awards and get a lot of additional grant money for doing that. So here he is, and this is my mark that I made. I went back and found my notes in my book from the meeting on my shelf in my office, and I clearly underlined his talk to go to that morning, Monday morning at the meeting. And so basically what he talked about was, although it wasn't that clear in his title, but he talked about the development of the infectious cDNA clone for hepatitis C. Basically, this was the result that was the $1 million or however much he got for the Nobel Prize result. So this is basically showing that you know, in the they were measuring hepatitis C RNA in the infected chimpanzee, and they could, you know, follow the RNA going up in concentration. So it was, you know, the virus was being made and circulating in the blood, and so they could see the presence of viral RNA. In addition to that, they checked for some liver enzymes, and basically when you have hepatitis, you start damaging the liver, and so you start to see an increase in liver enzymes, and they were the two different ones here, and had a peak, you know, a pattern for the production of that above the, you know, the normal baseline. And so this was a figure in the science paper as well. And just for fun, I went back and looked at my notes, and actually I had, I think, 8 pages of notes from his talk. And so here was my, and I'm usually really that good at taking notes, but here's my diagram, you know, for what he was showing here and here. And so here's the, you know, the diagram for the RNA going up, and here's the enzyme, increase in enzyme activity. And then he said he got, you know, got a second animal, and that's over here. I'm sorry. Second animal got a different pattern. The 5' in other words, whether you modify the 5' end with additional bases or the 3' end, it didn't really matter, like we talked about before. And this really set the stage for a lot of, you know, future studies. So I was impressed that my diagram was recognizable actually compared to the papers, you know, the figure. And I'm sure this is the figure he showed at the meeting. Anyway, so it was just a, you know, one of those times when you get to hear impressive scientific work. You don't know that it's going to be Nobel Prize winning work, but you kind of think it might be. And so I got, you know, I was, this was one of the examples where I got to hear it firsthand when I, you know, it was talked about before the publication came out. I was also, when I was at MIT, I got to hear Phil Sharp give his talk on splicing from his lab that eventually won him a Nobel Prize as well. All right, so to wrap it up, in terms of conclusions, the HCV cDNA clone containing the consensus sequence was infectious. And so, you know, that was what was needed, and that just led to all kinds of follow-up work. Actually, some work that was done in my lab, we ended up, you know, having a copy of that cDNA clone, and we were, you know, looking and expressing and had a collaboration with a company that Charlie also collaborated with, trying to develop RNA polymerase assays. And so we actually made some progress on that, did that for 3 or 4 years. And so, you know, the clone was extremely valuable to a whole lot of people. The new HCV viral RNA produced, he showed that it was actually packaged. It wasn't just RNA. And so, in other words, all the RNA was RNase resistant. It was inside of the virus. The sequences at the end seem to be a little bit flexible. The additional, you know, 5' terminal and 3' terminal sequences were tested, but it didn't really matter. All those clones were infectious, even though there was a little variation there. Sorry. Anyways, the bottom line was the results proved that HC viral genomic RNA alone was the sole cause of the disease. So that really went back to the discovery in 1989. You know, that was the final piece of information that was really the ultimate proof. In other words, that the RNA, the genomic RNA from HCV virus, was able to, in and of itself, be the sole cause of hepatitis in people infected with hepatitis C virus. And then the isolation of the infectious clone in the Rice lab led to the development of new cell culture systems. Once they had the infectious clone, they knew it was infectious, so then they could search for cell culture systems, by modifying those clones in some ways. I mean, actually, they set up a way to select for cells that could support, virus replication. Anyway, over time, they developed really good cell culture systems to study viral RNA replication and express, the viral enzymes. And this led to the development of new and effective drugs, that were used for a treatment of HCV. So that's it. I'm happy to, answer any questions if anybody has any.
We can go back to the gallery.
Say that again.
I said we could go back to just the gallery view of the people here, unless you want to leave that slide.
Okay, sure.
Question.
That's a good idea.
Ken, do you want to handle the question?
Ken, you need your microphone.
I'd be happy to. I don't mind.
I'm fine. We're ready to go. You have a question that you want to ask, John?
Yeah, I have a couple of interesting questions. Those 34 clones that they tried on the chimps, was it just bad luck that they didn't have the consensus sequence among those 34? And had it worked, would it have sidetracked the research? I'm just wondering.
Yeah, I mean, statistically, you could have predicted it once you knew everything that was going on. But there would be clones in there that were, there were RNAs in that population, but, that would be infectious, but a lot of them weren't. And so, when you pick it's really a small number compared to almost 1 million, RNA molecules
Right.
In that population. So, and they're just, they get picked at random as far as we know.
Yeah.
And so, yeah, so they had to, it was, they could have been lucky and gotten one.
Yeah.
That's a good question. And the quasi-species issue, once they, and I don't think we fully appreciated, there's been a lot of work, done on that and looking at RNA virus evolution since, 1989. So we have a much better understanding of what's going on now than we had then, with all the new sequencing techniques, deep sequencing capability. We were just starting to scratch the surface then.
One other question, and then maybe someone else will have some questions. Can you put, this sounds to me like you could make a mistake and produce some new diseases that man had never seen before. I'm just wondering the Frankenstein effect of fooling around this way. When you use restriction enzymes and put together something new, you might find yourself suddenly embarrassed. I'm just wondering if that's a possibility.
So that is a possibility, and people in the field worry about that. Ken has in fact been on committees where they definitely worried about such things, happening.
Mm-hmm.
But in it, basically an example that's pretty scary in a way is basically using slightly different techniques, but we've reconstructed the 1918 strain of influenza virus to study and learn why it was so pathogenic. The take-home message there is the virus itself is very pathogenic. I mean, with flu, it's a respiratory infection. You can get secondary bacterial infections. We didn't really have, the whole array of treatments for bacterial pneumonia back in those days. So there was always kind of a question as how pathogenic was the virus. It was very pathogenic. And so, and the sequence is published. And so, you know, anybody that knows how to do, you know, not just anybody can do it, but I mean, somebody, any of our graduate students in a molecular biology lab could do it. You know, could reconstruct that from the sequence if they had the, you know, the equipment and supplies and so forth.
So before going to a question from Ken, can't they develop a vaccine now to the 1918 influenza, just in case we might need it?
You could. I mean, we know what the capsid proteins are and so forth. So in principle, we could, you know, have a, we could in principle do that.
Warp speed. Yeah. Ken.
It turns out, of course, John, that the virus is always smarter than we are. And so by the time you got your vaccine, it would have mutated to escape your wonderful vaccine. I mean, that's one of the issues. I spent 8 long years on the recombinant, well, 4 years on recombinant DNA advisory committee, and 8 years on the National Scientific Advisory Board for Biosecurity. And that's why those committees existed. Were we going to create something that would cause a terrific pandemic and wipe us all out? And, you know, the people that argued that that could happen were sort of looked at with suspicion. But the good the real virologists, these were all people who were theoreticians for the most part. The real virologists said, look, there's plenty of good stuff out there. You know, if you're going to have a pandemic, it's going to be something that nature will do for you. And one only has to look at what's happening now to see that. But interestingly, of course, one of the conspiracy theories is that the laboratory in Wuhan was working on the virus as it was, and that somehow there was an escape of the virus from that lab, which got out into the community and caused the pandemic. And all the studies that have been done say that's unlikely, but you cannot 100% rule out that possibility, especially since the Chinese government has not been totally forthcoming with all of the early information about the pandemic in Wuhan.
Right. Barbara has a question, unless you had a question. Barbara?
Yeah, my question is along the lines whether the types of committees that Ken sat on, sort of like control groups, more or less, or review groups, in places like China and Russia and other major areas where they have this type of research, do these foreign governments have similar types of review or control groups that you know of?
Yes, they do, but you don't know what they're really doing. I mean, 1972, we signed a convention outlawing biological weapons, and everybody signed on to that, including especially the Russians. In 1990, with perestroika, we discovered that the Russians were making literally tons of smallpox every year to weaponize, to use as biological weapons. So I've become a skeptic in some respects, and of course we never had an inspection regime with the convention until recently. But in 1990, when things opened up, I saw pictures the CIA took of these underground laboratories that were really scary. You could drive semi-vans through them, and the fermenters were huge, et cetera, et cetera. The problem is that for warfare, nature has provided us plenty of good ammunition, unfortunately. Now, I think one of my other committees, I keep getting stuck on these things. Once you're on one, they come after you. But you may remember a few years ago that smallpox, which is not supposed to be anywhere except in 2 labs in the world, was discovered at the NIH in a cold room in an unsecured situation. 50 years old, these cultures were. And once they discovered it and somebody owned up to the fact they were there, they immediately got whipped off to the CDC, who tested them. And it turned out that after 50 years at 4 degrees, which is, you know, refrigerator at best, that several of these cultures are still biologically active. So this is the problem that we face when we try and set up a regime where we control who has cultures of these organisms, and we try to make sure that there are no accidents, that they get tracked out to the outside. And it's, you know, when we had the anthrax in 2001, even though only 6 people died, we know what panic there was. But the biology is variable. You just don't know. I mean, Bert described the fact that about mutation, you know, the best vaccines against polio were live attenuated vaccines, okay? And they were great. I mean, they were very effective. And they prevented people who got immunized from spreading the virus, as well as protecting them. The only trouble was that when this live virus vaccine multiplied in the GI tract of the people who got it, turned out within a couple of days, many of them were putting out virulent or paralytic poliovirus by mutation. And today, and I'm on another committee about that still, we worry about polio, and wild type polio is gone almost totally, with just a couple exceptions in Pakistan and Afghanistan. But there is endemic polio around, and all the endemic polio that's around is derived from the live virus vaccines, and it gets spread in the community, and you still have to worry about it. The result is that the World Health Organization has now banned the use of live virus polio vaccines. So.
Okay.
John?
Yeah.
This is Julianne. I have John Axe here in the Acorn Room. He has a question.
Oh, great. Good.
John, thank you. Thanks for the presentation. I'm a physicist, and I know a little bit about how physics prizes are awarded, Nobel prizes, and I suppose every committee has similar kind of things. This work was done 20 some years ago, and in physics they would have a long list of names of people who've been suggested for important work, and every year they go through this. And in physics recently we see a lot of a sudden realization that this person is getting close to 95 years old. And since we can't give a Nobel Prize posthumously, it's now or never for them. Does that happen in here also? And is that part of why this year these people got the Nobel Prize?
Could you hear that okay?
Yeah.
I'm not quite sure what you wanted. Was he wanted to know whether they do it just because this is their last chance?
It's those committees are secret discussions about these things. I'm sure that in the Swedish Academy and that sort of thing. But there have been some superannuated awards recently. Someone in their late 90s or mid 90s won a few years ago. I can't even remember the name of the person. I don't think they take that into account, John, but I don't know. And maybe someone else knows. I'm pleased to see that next week's lecturer is with us today.
Yes, it should be announced that he has a lecture just preceding this session, a university lecture. He's promised us that he's going to try and get here as close to the starting hour as possible. So I think, I don't know if even in the announcement we gave him an extra 15 minutes or not, but be patient next week because the topic, which is where we can edit genes. I mean, you're really in your genome on the spot is really quite extraordinary. This is one of the no-brainer Nobel Prizes. Nobody was surprised. The only question was how soon would the two discoverers actually get it?
So they're young people. Julianne.
We have one more here from the Acorn Room.
Oh, okay.
Hi, Bert. This is David Challoner.
Hi, David.
Thank you. We all know just about the sociology of science in various arenas. Who has their nose out of joint if they didn't participate in this prize?
I didn't quite catch the question.
He was asking, were there people upset that they didn't get part of the prize?
Correct.
It's always limited to 3 people.
So I think that just some of the articles I read, about the announcement and the people, the different groups, I think, there was clearly some of that here as well. The 3 people were the senior people in each of the 3 different, groups. And then I think in particular for the, work that was done in the initial, clone that was developed in 1989, where they, identified it as a virus that was an infectious agent. I think there were, other members of the group that had worked, very hard on that for quite a while. And so I think there was some, but it wasn't like a separate group, that didn't get it, but I mean, they were limited to 3 people. So I don't know if you're going to follow, those rules. I think it's going to happen, but I think that does happen, in some cases, certainly. I mean, there's some other, ones that I think some of us could tell stories about, splicing, for example, would be one that I know.
There's the talk next week. There was a second group in Boston that did some very early work applying the results of the discovery of the CRISPR and Cas enzyme. But it was very interesting. The lead guy in that is a guy named George Church, who's a certified genius. And they said, did you feel that the 2 women, Jennifer Doudna and Charpentier, getting the prize was unfair, that you should have had part of it? And he said, no, they discovered it. We figured out, we think, how to apply it first. But that's the prize is for discovery, not for application. And I thought that was a beautiful answer and right to the point.
Okay. Any further questions? Then we'll see all of you next week.
That's all.
Yeah, stay tuned.
Flanegan.
You're welcome. Thanks.